Welcome everyone. In my previous video, I have discussed about how Ernest Overton in 1895 discovered that biomembranes are chemically lipid in nature. Now in this video, I will talk about how scientists discovered that these lipids are actually structurally bilayer in nature. Now for this discovery, two scientists, E. Gorter and F. Grandel, in 1925 performed one experiment. Okay, they took red blood cell. Now why red blood cells are taken or chosen over other cells? Because mature red blood cells lack both nucleus and cytoplasmic or cytosolic organelles. So the plasma membrane is the only lipid containing structure in the red blood cells. So if you extract, now they extract lipids, from the red blood cells. So the lipids are actually coming from plasma membrane. So that's why they have chosen red blood cells, because mature red blood cells don't contain nucleus or other cytosolic cellular organelles. Now extraction methods is very simple. They used organic solvent. Like hexane. Now when they evaporate the organic solvent or hexane, they get the lipids that is extracted from the red blood cells. Now they used one apparatus or instrument that is called Langmuir trough, TROUGH. So they use one instrument. This instrument is named upon its inventor, Langmuir. So they used Langmuir trough. I will show the image of this instrument. This is the image of Langmuir trough instrument. E. Gorter and F. Grandel used this instrument for the experiment. They extract lipids from the red blood cells and spread the lipids over the water. And this is the movable barrier that can compress the lipid molecules to the smallest possible area. Now you can measure the surface area of water covered by lipid molecules, and if you divide this surface area with the surface area of the red blood cells, you will get the ratio that is very much close to 2:1 ratio. That means lipids present in the plasma membrane in a bilayer manner. The instrument looked like this. This instrument actually has water. Let's suppose this is the water, and here is a movable barrier is present that can move. So this is the movable barrier that can move. Now when they put these extracted lipids on this water, the lipids actually spread over the water. And we know that if we put oil on the water, there must be some gaps in between the lipids. So we can actually move these movable barriers to actually compress the lipid molecules to a smallest possible area where all lipids present. So we can move these movable barriers, and by this way the lipids can be compressed, and there is no gap in these lipids. You must remember, these lipids are actually layered as a monolayer, not bilayer. This is monolayer, because lipids, when dissolved or present on surface or over the surface of the area, they actually form monolayer. So this is monolayer lipids, and let's suppose here is the movable barrier. So you can compress the lipids so that it can take the smallest possible area. What is smallest possible area? Okay. Now they actually measure this surface area of the water, but they did two mistakes, and these two mistakes are also important to get into that result. The mistakes are, first mistake they perform, that is incomplete extraction of lipid from the RBC. And second mistake is underestimation of surface area of the RBC. So they did two mistakes, and these two mistakes actually nullify each other. They extract the lipids from the RBC, so but incomplete extraction occurs, not complete. And also they estimate the surface area of the red blood cells, that is less, so underestimation of surface area of red blood cells. Now they make a ratio. So these two mistakes I can write here, these are the mistakes they performed, mistakes, and these two mistakes led to the correct answer. They actually calculate the ratio of two surface area. First surface area is this, the surface area of water covered by extracted lipids. Okay, they make a ratio, they calculate the ratio, the surface area, this area. Okay, so they calculate the surface area of the water covered by extracted lipids, and they divide this surface area with the surface area of red blood cells. Now, as I have mentioned, they did two mistakes: incomplete extraction, so there must be less surface area of lipids, and also they underestimate the surface area of the red blood cells. So both these mistakes nullify each other, and they conclude, or they actually lead to the more or less correct answer, that is, this ratio comes about 1.8:1, and sometimes they got 2.2:1. That is very close. This ratio is very close to 2:1. That means if you take the surface area of RBC, you will get double surface area of the lipids that actually cover the water surface. So as this is the monolayer, so there is double of the surface area in case of when we extract these lipids. So from this experiment, E. Gotter and F. Grandel in 1925, from this experiment they concluded that these lipids are actually bilayered in structure. Now this is indirect evidence, but the direct evidence comes when in the late 1950s, when J.D. Robertson, another scientist, took the image of red blood cells by electron microscopy, because electron microscopy, this instrument was discovered in late 1950s. So J.D. Robertson, another scientist, J.D. Robertson, in the Duke University, he took the first image of red blood cells by electron microscopy. He used one stain chemical called osmium tetroxide. Osmium tetroxide. This osmium is actually a heavy metal that can bind preferentially to the polar head groups of the plasma membrane, and I will show the image or the diagram, the photo that he taken from the electron microscopy. This is the electron microscopic image taken by J.D. Robertson. He stained the red blood cells with osmium tetroxides, and osmium tetroxide, the metals osmium, actually binds preferentially to the polar head groups of the lipid molecules. As you can clearly see, there is two layer of lipids present in the plasma membrane, and it definitely reveals that plasma membrane is lipid bilayer in structure. Hope this video will help you. Thank you for watching, and always keep learning.